I think we should get started. And good afternoon. My name is Yi-Hong Ye, and on behalf of the Protein Trafficking Interest Group, I'm delighted to introduce today's seminar speaker, Dr. Tom Rappaport. Dr. Tom Rappaport has a very distinct scientific career. He started his career in the previous East Germany, where he received his PhD training and completed the so-called habilitation in Humboldt University. He became a full professor in the Central Institute for Molecular Biology in Berlin in 1985, and later moved to Max Delbruck Center, which is also in Berlin. In 1995, he was recruited to the cell biology department at Harvard Medical School, where he has become a Howard Hughes Medical Institute investigator since 1997. For many years, Tom has been interested in a very fundamental biological question, that is how secretory and membrane proteins are moved across the membrane of the endoplasmic reticulum to enter the secretory pathway. His lab used an elegant cross-linking approach to define the components involved in these processes in bacteria and eukaryotic cells. He is also the first to reconstitute the richer translocation system using purified proteins and liposomes. And these experiments demonstrate for the first time that a conserved multi-spanning membrane protein complex encoded by the SEC61 genes in eukaryotic cells is the translocon or translocation channel that mediate protein import into the ER. He also demonstrated that the same protein complex can mediate the integration of membrane proteins into lipid bilayer. And in recent years, Tom's interest has diversified quite a bit. He has made several important discoveries on a process that transport misfolded proteins out of the ER for elimination by the ubiquitin proteasome system in the cytosol. He also discovered a first cellular factor that gives the endoplasmic reticulum its unique network shape. Because of his distinguished scientific contribution, Tom has won many prestigious awards and was elected to the National Academy of Science in 2005. In addition, Tom has been widely regarded by the scientific community as an outstanding mentor. He just received the Clifford Bagger Excellence in Mentoring Award, which is a very competitive mentoring award at Harvard University. And I was very fortunate to have the opportunity to work in Tom's lab as a postdoc a few years ago. I should also mention that Tom visited NIH a few years ago and talked about protein translocation. And after his seminar, he was asked by an audience how SEC61 complex could mediate the integration of membrane proteins into the lipid bilayer, as well as moving them into the ER lumen. Tom admitted at that time that the question could not be answered until we know the X-ray structure of the SEC61 translocon. Now Tom not only determined the structure of the bacteria translocon complex SecYEG, his group also recently captured the translocon in action by solving the structure of SecYEG in complex with its ATPase partner SecA. In my opinion, this is the hardest heroic achievement, given that SecYEG is a membrane protein complex that contains 12 transmembrane segments, which make the proteins very difficult to purify, let alone to crystallize. With these structures, I'm sure that Tom will have no problem answer the previously unanswered question, as well as many questions that have plagued the field for many years. I'm really delighted that Tom can come to NIH and share with us his most exciting findings. Please join me and welcome Tom to NIH. Thank you.
Okay, well, I don't know. Otherwise, I just use this microphone. Works? Okay, all right. Well, thanks for the invitation to come here. It's actually the second time that I have this big honor to give this type of lecture. Yi-Hong told me that he actually looked at the video that they made from the first lecture, and I'm afraid he's going to check whether what I said the first time is actually still true. And I believe it's probably not. You know, obviously, when you move on with science, then things turn out to be wrong. But I hope at least some of this, what I said, is still true. It's a particular pleasure to be hosted by Yi-Hong, who was a postdoc with me and really did some fundamental discoveries in the retrotranslocation system, which unfortunately I don't have time or even any new data to talk about. So what I will talk about today is the mechanism of protein translocation across, now we have another problem. Something wrong here with the, I see it on my screen, but unfortunately there's something wrong here. Here we go. Nope. Okay. Don't touch anything here?
Take it out of sleep mode for me?
What is the sleep mode here?
I do.
Okay.
I apologize, everybody.
Are you looking for the, I think it might be here. Well, as long as the audience isn't sleeping, I think it's going to be okay. Let's just go on.
I can't get it
Okay.
off of sleep.
All right. Just go back. Okay. Well. Okay. All right. Sorry for that. So this is what I'm going to talk about, protein mechanisms of protein translocation across membranes. And specifically, I'm going to talk at the major part of my talk is going to be about this new structure that Yi-Hong mentioned briefly, namely the structure of a SecA-SecYG complex. But I realize that this will not mean a lot to you, so before I do that, I'll give you quite a bit of introduction and kind of an overview about the field as well. So let me start out by reminding you that protein translocation across the endoplasmic reticulum membrane is really the first and decisive step in the biosynthesis of a large class of proteins, which includes secretory proteins, plasma membrane proteins, proteins of all organelles along the secretory pathway. In all these cases, proteins are initially targeted to the membrane by virtue of hydrophobic sequences, which can be cleavable signal sequences, as in the case of most secretory proteins, or non-cleaved transmembrane segments, as in the case of most membrane proteins. Subsequent to the targeting phase, proteins are moved through the membrane through a protein conducting channel that has an aqueous interior. In the case of soluble proteins, which include secretory proteins, they would move all the way across the membrane and end up on the other side of the membrane. In the case of a membrane protein, when a hydrophobic segment, shown here in yellow, arrives inside the channel, it would move sideways out into the lipid phase, generating luminal portions of the membrane protein as well as cytoplasmic domains. So this channel has a very unusual property in that it can open in two directions rather than only in one direction, like channels would do that transport small molecules like ions. Those channels only open perpendicular to the plane of the membrane to let something across, but this channel has ability to open perpendicular to the membrane to let a protein across, but also laterally within the membrane to let a hydrophobic sequence out. And this is something that we find particularly intriguing and like to understand how this works in molecular terms. Now it turns out that translocation in bacteria and archaea works essentially by the same mechanism. Again, we have signal sequences or transmembrane sequences. In the case of bacteria, there is no internal membrane, at least in most cases, and proteins are transported across the plasma membrane and end up, in the case of gram-negative bacteria, in the space in between the two membranes, which is called the periplasm, or in the case of gram-positive bacteria, you transport it across the plasma membrane and end up in the medium. So it turns out that the signal sequences and the transmembrane segments look principally the same as in eukaryotes, and the mechanism of translocation is also similar. So the channel that is involved in translocation is in fact evolutionarily conserved. It's a heterotrimeric membrane. It's formed from a heterotrimeric membrane protein complex that is called the SecA-mediated complex in eukaryotes and SecY complex in bacteria and archaea. The conserved subunits are the alpha and gamma subunits, which are shown here in red. And those are also the two subunits that are essential for viability. So in cases where you can test this easily, like yeast and bacteria, if you delete these alpha subunits or the gamma subunit, you're dead. The beta subunit is neither essential nor is there universal conservation. Now, before I go on, I'd like you to remember the names for the components in bacteria. Unfortunately, the names of these components are awkward, but they're traditionally determined, so I cannot change them. But because I will use the names for the complex in bacteria, I'd like you to remember those. So the alpha subunit is called SecY, beta is SecG, and gamma is SecE. The original evidence, as Yihong was already mentioning, for this complex forming a channel came from photo-crosslinking experiments and from reconstitution experiments that we did almost now 20 years ago. So the components, the topology of these components is such that the alpha subunit spans the membrane 10 times with N and C terminus in the cytosol. The beta and gamma subunits in most organisms are single-spanning proteins with the N-terminus in the cytosol. Some beta subunits have an additional transmembrane segment, so for example, it's a SecG protein in bacteria, and also some gamma subunits have additional transmembrane segments, such as the SecE subunit in E. coli, but those are actually non-essential. Now, the important point is, and this is, I cannot stress this strong enough, that this channel is only a passive pore. It doesn't transport proteins on its own. In order to do so, it needs to associate with a partner that provides a driving force. And we know of 3 different ways of how the channel can be put to work, and those 3 ways can be distinguished into 2 different modes. So the first mode is called co-translational translocation. It's the most general mode that is found in every cell, in every organism, and it's also responsible for the integration of most membrane proteins. So in this case, the channel shown here in red associates with the ribosome as the major partner. The ribosome sits on top of the channel such that the tunnel inside the ribosome is aligned with the pore in the membrane. And then as the polypeptide chain is elongated by translation, the polypeptide chain moves from the tunnel in the ribosome on into the channel and then on into the lumen of the ER, or in the case of bacteria, to the outside of the cell. So the principle here is that the polypeptide chain is only one way to go. And the energy for translocation essentially comes from the energy of translation, which is GDP hydrolysis. All cells also have a post-translational mode of translocation, very likely. So the fact that you have those 2 modes coexisting in the same cell obviously raises the question of when do proteins take the co-translational mode and when do proteins take the post-translational mode? And the answer to that is that the more hydrophobic signal sequences take the proteins to the co-translational mode. Post-translational translocation, which means that the polypeptide chain is first completed in the cytosol and then translocated, occurs differently in eukaryotes and in bacteria. In eukaryotes, the same channel associates with another membrane protein complex, the SEC62-63 complex, as well as with a luminal protein called BIP. BIP is a member of the HSP70 family of ATPases. It's a chaperone as well, and it acts as a molecular ratcheting molecule. It binds to the incoming polypeptide chain and then prevents the polypeptide chain from sliding back into the cytosol, simply because the bound BIP molecule is too large to fit through the pore of the channel. However, the chain can move in the forward direction, and then the next BIP molecule will bind, and so on, until the polypeptide chain is completely across. So we call this a ratcheting mechanism, and because the polypeptide chain is sliding back and forth by Brownian motion, this is also called a Brownian ratcheting mechanism. And then finally, in bacteria, this mechanism wouldn't work because there's no ADP on the outside of the cell. So instead, we have an ATPase on the cytosolic side called SecA, which uses the energy of ATP hydrolysis to push the polypeptide chain through the channel. So the important thing here is that the channel is essentially always the same, the partners are different, and the mechanism by which the driving force is provided is completely different in the three different modes of translocation. So it's a very interesting modular system. In this talk, I'll be concentrating on this mode of translocation that we have in bacteria, and until recently, until the structure that we obtained last year, we really had no clue of how SecA would push the polypeptide chain through the channel. Now we at least have some ideas. Before I talk about this new structure, let me just talk a little bit about the channel. So 5 years ago, we've been able to solve the first structure of the first protein-conducting channel. This was work from 3 postdocs in the lab, Bert Vandenberg, Bill Clements, and Ian Collinson, and was a collaboration with Steve Harrison. So this shows the channel. You would sit in the cytosol looking down onto the membrane. These squiggles are the transmembrane segments of the alpha subunit of the protein. So in color is the alpha subunit. The alpha subunit is divided into 2 halves, transmembrane segments 1 through 5 in blue and 6 through 10 in red, and they're linked by a loop between TM5 and 6 on the extracellular side, which we call the hinge. The 2 halves are kept together by the gamma subunit, which wraps around the 2 halves. There is a helix that lies kind of flat on top of the membrane, and then there is a very tilted transmembrane segment of the gamma subunit. The beta subunit sits on the side of the alpha subunit, makes relatively few contacts, which may be an explanation of why this polypeptide chain is not essential for translocation. Now, the important point is that this molecule can open up sideways on this side, which we call the front or the lateral gate. So basically, this molecule can open up like my fingertips do here, which obviously immediately suggests the mechanism of how transmembrane segments would be released from the channel into the lipid phase. I always have these kind of cartoons in the corner here, which may perhaps facilitate to understand what I'm talking about. Now, this shows a space-filling model of the channel, and these 2 lines indicate where the border of the membrane would be. And as you can see, there is a funnel on the cytosolic side, but it tapers to close approximately in the middle of the membrane. There's also an extracellular cavity, but there's no connection between the 2. And so this is a closed state of the channel that would not transport even small molecules. We've shown this by electrophysiology experiments, let alone proteins. And this is, of course, not a surprise because we crystallized the channel without substrate and without channel partner. So this is a closed state of the channel. So basically, we have to guess how the channel would be open for translocation. And that's actually quite easy to do. All you have to do is color the molecule correctly. So if you look at this thing here, if you think away this green helix, then there would be a hole in the center of the molecule. So for obvious reasons, we call it a plug. So the idea is then that the plug would move out of the way, as shown here, and this would open the channel. This is not just speculation because both Tom Salhavi's and Frank Duong's group have put a cysteine into the plug and a cysteine into the gamma subunit and shown that you can form a disulfide bridge, whereas in the closed state of the channel, the 2 cysteines would be too far apart to form a disulfide bridge. Now, this opening of the channel doesn't occur spontaneously, of course. You need a trigger, and the trigger is the signal sequence. So the signal sequence would intercalate between these 2 helices shown here in color, in blue and in yellow, and the hydrophobic part of the signal sequence would form a little helix of perhaps 2 or 3 turns, which would be hydrophobic all around. And then this helix would intercalate between these two. These 2 helices would separate, as shown schematically down here, and then the helix shown here schematically as a cylinder would intercalate between these 2 helices of the channel. This would destabilize interactions that keep the plug in the center of the molecule so they can move out of the way and open the channel. So once you've done that, you convert the channel into an hourglass shape. So not only you have a cytoplasmic funnel, you would also have a funnel on the extracellular side, and there would be only a small constriction in the middle of the membrane between these two funnels. Turns out this constriction is formed by 6 hydrophobic amino acids that project their side chains radially inwards. In E. coli, all 6 amino acids are isoleucines. We call this the pore ring. So the idea is then that the polypeptide chain would move from the cytoplasmic funnel through the pore ring and then on into the extracellular funnel and then on into the lumen or extracellular space in bacteria. So there's a few points I'd like to make here. First point is that the polypeptide chain would move in an aqueous environment all the time. Basically, the barrier of the membrane is confined to a single layer of side chains here. Second point is that the pore ring would prevent other molecules from passing through the membrane during translocation. If you think about it, it's not a trivial thing to allow a macromolecule to go through and at the same time prevent small molecules from moving through the membrane. If you think about E. coli, for example, there's a membrane potential across the inner membrane, which is the major energy source for the cell. So you don't want to dissipate the membrane potential while you're moving a polypeptide chain. The idea is then that this serves like a gasket around the polypeptide chain, preventing the free flow of ions. It's not likely to be a perfect seal, but at least it would prevent the free flow of ions. Finally, the hourglass shape and the hydrophobic nature of the pore residues would minimize contact of the polypeptide chain. And this is also an important point because you want to minimize friction of the polypeptide chain while you're moving it through the channel. The final point on the structure is that, as I mentioned already, if a transmembrane segment arrives inside the channel, it will move out sidewise into the lipid phase, as shown in the, schematically down here. Turns out this lateral gate is actually pretty complex. It's made of 4 little segments of 4 different transmembrane segments, and so we believe that this seam is pretty weak, so it can open up pretty easily. Now, I hope everyone realizes that these are just speculations based on the X-ray structure, but still speculations. And so ever since we got the structure, we set out to test some of these ideas. And the first question we had, is it really true that the polypeptide chain moves through the center of a SecY channel? The reason why this is an important question is because before the X-ray structure, we and basically everyone else in the field thought that the channel would be formed by a ring of several Sec61 or SecY complexes that would form an oligomeric assembly with a hole in the middle of the assembly. So this obviously was a very different model. So the way we tested the prediction from the X-ray structure, and we meaning Kurt Cannon and Aaron Orr, 2 postdocs in the lab, is by putting a cysteine into a translocation substrate and then another cysteine at many different positions of the SecY molecule, and then ask the question whether you can form a dicipher bridge. If so, you would say that this cysteine is in the path of the polypeptide chain as it is moving through the channel. To make a long story short, what we found is that the green dots give you dicipher bridge formation, and the red dots don't. And the red dots were chosen at the circumference of the SecY molecule to test this alternative model that we favored before. What's really remarkable is that if you look at the space filling model and look where the green dots are located, it turns out they're all in the waist of the hourglass and the waist of the channel, the narrowest constriction. And so there's 2 conclusions here. First, the pore is in fact located in the center of the SecY. And second, contact of the polypeptide chain is in fact confined to the channel constriction. Now this does not necessarily say that the channel is only formed from one SecY molecule, because what you can imagine is that you have 2 SecY molecules coming together, opening their lateral gates and fusing into a larger channel. You would still have the same residues facing the polypeptide chain. So the crosslinking experiments don't address that. However, there's other evidence to suggest that the pore is in fact formed by just one SecY molecule. And part of I'm not going to show you the data that were obtained by Andy Osborne, also a former postdoc in the lab, but he showed that both the signal sequence and the mature region of a translocation substrate are contained in the same SecY molecule. This is, again, disulfide bridge cross-linking. Furthermore, we have some evidence from electron microscopy, and these are experiments that we did in collaboration with Chris Akey at Boston University. So what these experiments, and what we do in these experiments is simply the following. We take purified ribosomes, purified SecY complex, and mix them together so they form a complex, and we put them on an electron microscopy grid so they land on the grid in all different orientations, and we get different projections of the same three-dimensional object, and then we do a reconstruction. And so what you get is something that looks like this. So here's a small ribosomal subunit, the large ribosomal subunit, and down here is the channel. Now, the important point here is that the volume of this channel is sufficient for a monomer of SecY complex. And in fact, the resolution is now good enough so that we can dock the X-ray structure into the density. So we're certain that there's just one SecY molecule bound to the ribosome. Now, these are non-translating ribosomes, I have to stress. And we get similar results also with the eukaryotic ribosome and Sec61 complex. So it's, again, just one copy. Now, the important point here is that this one copy sits on the ribosome in such a way that it sits right on top of the exit site of the ribosome. So this white dot here is the site where the polypeptide chain would emerge from the ribosome towards you. And as you can see, the channel would sit right on top of it and could accept the polypeptide chain when it comes out of the ribosome. Now, as I said, these are non-translating ribosomes, so there's a caveat here. Obviously, we're very interested in getting a structure of the ribosome nascent chain channel complex. We've been trying this for some time, and hopefully we'll get it at some point, but so far we don't really know for sure. But at least this is consistent with the idea that just one copy of this Sec61 or SecY complex would form the channel. So there's one important consequence of assuming that the channel is only formed from one SecY molecule, namely that the pore size is relatively small. These are actually the maximum dimensions that you can imagine. If you stretch the molecule in the computer, you might be able to envision a channel that is 20 times 15 angstroms in dimensions. But it's certainly much, much smaller than 40 angstroms diameter that other people have proposed. Now, here's a big surprise. So despite the fact that you only need one copy to form a channel, it still looks like you need oligomers, at least for the SecA-mediated translocation, to mediate translocation. And the evidence for this comes from experiments in which we used so-called tandem molecules. This is, again, work from Andy Osborne. So tandem molecules were introduced by Frank Duong, and basically what they are is you fuse two SecY molecules at the gene level such that the C-terminus of the first SecY is covalently linked with the N-terminus of the next SecY molecule. And then you introduce a mutation in one or the other molecule to kill the molecule for translocation. And then we ask the question whether you can get translocation through the dead copy if linked covalently with a healthy copy. In other words, does this wild-type copy rescue the mutation in this copy? And the simplest interpretation of such a result would be that oligomers mediate translocation. Now, how do we know whether a translocation occurs through this or through this copy? Well, the way we do this is by introducing a cysteine into the substrate and a corresponding cysteine in one of the SecY molecules, and so we can test whether translocation occurs through this or through this copy by using disulfide bridge cross-linking. Let me give you an example of this kind of experiment. So what we're doing here is SecA-dependent translocation. Therefore, we only see cross-linking when we have ATP around, simply because you don't see translocation in the absence of ATP. So if we do cross-linking experiment with a healthy monomer, you see that you need ATP to see a nice cross-link. If you kill that copy with a mutation, you no longer see this cross-link. The rest are tandem molecules. So if you have two healthy molecules, you get cross-linking. The cross-link product migrates is a little bigger, so that's why you see a shift here. If you kill both copies, there's no cross-linking, and here's the important experiment. So here we're looking whether you get translocation through this copy when you link it with a healthy copy, and as you can see, you get cross-link. These two lanes are just simply the other way around. So the conclusion would be you need oligomers for some reason. So why is an oligomer needed if only one copy is required to form the channel? So the model that we have is the following, that the SecA molecule would interact with one domain with a non-translocating SecY copy and would move the polypeptide chain through a neighboring copy. You can think of it like a static interaction. So let's say you have a static interaction with one SecY and you would push it through the other. And the idea is that if you didn't have that static interaction, every time the SecA molecule resets, it would fly away. So you might have to have some interaction. So in order to test this, Andy did a really heroic experiment. He looked for, as pair of cysteines, one cysteine would be in the SecY molecule and another cysteine in the SecA molecule that would be able to form a disulfide bridge. And after 1,000 experiments, he found a pair, one cysteine in the MBD1 domain and another cysteine in the 6-7 loop of SecY that would efficiently form a disulfide bridge. So now we can do the experiment. So we're using, again, tandem SecY molecules, and we can ask the question whether the polypeptide chain is moved through this SecY molecule or through this SecY molecule. Because we suspect that this is the non-translocating, the interaction with a non-translocating copy, we would expect that you can only cross-link to this copy. And if you put the cysteine that would be able to cross-link with the substrate into this copy, you wouldn't see a cross-link. And this is, in fact, what we see. So without going through all the controls here, this is the important cross-link here, where we cross-link the nascent chain, the polypeptide chain, with the tandem construct and SecA this here. Whereas if you put the cysteine in the other copy, there's no cross-linking. So the conclusion here is that the MBD1 domain, MBD stands for nucleotide-binding domain 1, and I'll come back to this in a minute, binds to the non-translocating copy and pushes the polypeptide chain through the neighboring SecY copy. This is a very interesting and not common dimer, I have to stress that. We knew, of course, about channels like the potassium channel, where 4 identical subunits come together and the pore would be at the center of the oligomeric assembly. We also knew of situations like the trimeric porins, where you have a homotrimer and each of the subunits forms a pore. But here we have a situation where initially the 2 subunits are identical, but once translocation has started, one of the subunits is a translocating copy, the other one is not. And we think that this kind of non-identical dimers, which originated from a homodimer, may be more common among translocation systems. Perhaps the best system is the Usher system that is involved in the secretion of pili across the outer membrane. In this case, you have 2 outer membrane barrels, 2 beta barrels, and one of them is secreting the pili, and the other one has a different function. So again, homodimers, but once you start, one is the translocating copy, the other one is not. It's also possible that the channels that are involved in mitochondrial protein import, TIM40 and TIM22, where there's known to be twin channels that they'll act in a similar way. Okay, so now I'm going to switch to the newer data, the crystal structure of the SecA ATPase bound to the SecY protein translocation channel. And this is work mainly from a postdoc in the lab, Jochen Zimmer, with significant help from Yunsen Nam, also a postdoc. So before I show you the structure, let me first outline in broad terms how SecA works. So SecA might start in the cytosol by interacting with polypeptide chain, and then this complex binds to the channel. However, it's also possible that SecA binds to the channel first, and then the substrate joins. Regardless of whether you take this or this way, it's possible that both are taken. In the next step, you need ATP hydrolysis to transfer the polypeptide chain from SecA into the channel. And it will insert as a loop into the channel with a single sequence intercalated into the walls of the channel, like I indicated before, between these two colored transmembrane segments and the lateral gate, and the following segment in the pore proper. And then you need further rounds of ATP hydrolysis to move the C-terminal part of this loop through the membrane, as shown here, whereas the single sequence stays put. Eventually gets single sequence cleavage. So we believe that our structure would correspond to one of those two states because it's lacking a substrate. We have no polypeptide chain in the structure. So this is not a state where we expect the channel to be completely open, because for this state you need the single sequence. So this is not yet the open state of the channel. This would be the next stage of our long journey, I guess. So what we expect to see is something like a pre-activated state in which the channel is prepared for the arrival of the substrate but is not yet open. So we obtained structures, 3 different structures with SecA and SecY derived from different species. The best resolution we obtained was both components derived from Thermatoga maritima. The resolution is still not very good, 4.5 angstroms, but the maps are actually pretty good and the R3 is reasonable. So all these complexes were generated by simply mixing purified SecA with purified SecYG. Obviously, this is a membrane protein. You have to do the purification in detergent. And then we dialyze against ADP plus beryllium fluoride, which mimics some kind of an transitional state of ATP hydrolysis, presumably a state that is close to the ATP state. Turns out that all complexes have the same architecture. In each case, it's a one-to-one complex of SecA and SecYG. And this immediately tells you that we've presumably lost the non-translocating copy. I can tell you right now the copy that we see is what we believe would be the translocating copy if a polypeptide chain was present. Okay. So this shows the structure. The 2 lines indicate where the boundaries of the membrane would be. The gray stuff down here is SecY. The green stuff is SecG, the beta subunit, and the purple is SecE. So as I mentioned before, SecE has this helix that lies on top of the membrane, and then this is the tilted transmembrane segment of SecE. And on the cytosolic side is the SecA molecule. SecA is a flat molecule, and with its flat side, it lies kind of parallel to the plane of the membrane. This just shows the complex in its full glory, and there's nothing I can really say about this. Well, maybe except for the point that the only thing that comes close to the membrane is this N-terminus of SecA. It's been known for a while that SecA translocation requires negatively charged lipids, and unfortunately our structure doesn't give a clue of why that may be. This shows a top view. So again, you're sitting in the cytosol and you're looking down onto the membrane, and this is the SecA molecule, and the background would be the SecY molecule. So this serves to explain a little bit the domain structure of the SecA molecule. So SecA is a Rec-A-like ATPase. It has 2 nucleotide-binding domains, NBD1 and NBD2. The nucleotide is bound in between the 2, and these 2 domains move relative to one another during the ATP hydrolysis cycle. And this is where all the movement comes from that is required to push the polypeptide chain through the membrane. Out of NBD1 comes this yellow domain. So there's a beta strand, then there's this folded domain, and it goes back in. This yellow domain is called the polypeptide chain crosslinking domain, or for short PPXD. Following NBD2 is this domain in brown, which we call the helical scaffold domain. There's a long helix, and then there are 2 additional helices that belong to the same domain. And then finally we have this domain here, which is called the helical wing domain. The non-translocating copy that we lost during the purification would bind proximately here, and we know this from cross-linking experiments. So this would allow it to interact with the other copy in approximately back-to-back orientation. Most of the contact with the channel are done through this PPXD domain, but also this long helix lies kind of flat across the channel. We think that this may be a kind of a lever arm that would amplify the motion that you see here to the other parts of the molecule. The most interesting and important thing, we believe, is these two helices, which we call the two helix finger. There's a helix that goes in, then there's a loop, and then a helix that comes back out. And this helix pokes right into the cytoplasmic opening of the SecY channel. And as you will see at the end of my talk, we believe this is the most important feature of the channel, of the SecA molecule. All previous structures of SecA were in the nucleotide-free or ATP state. This is the first structure in the ATP state, but there's really nothing very special about this because all Reg-A-like ATPases do the same thing. What they do is that in the nucleotide and the ATP state, the two MBD domains move relative to one another 15 degrees. They close around the nucleotide, and there's an arginine finger that comes in from one domain, contacts the gamma phosphate, and this induces ATP hydrolysis. And this is true for all these ATPases. Here's more important and more interesting things. Both SecA and SecY channel undergo major conformational changes upon interaction. So let me first talk about the SecA changes of SecA. So on the left side, you see the structure that was originally solved by John Hunt in Hans Deisenhofer's lab, which is now called the closed state. In this structure, the PPX domain was closely opposing the helical wing domain. Then Andy Osborne in my lab solved another structure in which this PPX domain had moved away from the helical wing domain and opened up a groove here. And our initial thinking was that this is the groove where the polypeptide chain would bind. Turns out to be wrong. What actually happens is that PPX domain moves around all the way. These residues shown here in ball presentation stick in between the two nucleotide binding domains and come actually pretty close to the nucleotide binding site. The overall rotation from here to here is an amazing 80-degree movement. So it's a great 80-degree rotation. This shows just a movie which indicates this really dramatic conformational change. So we believe this conformational change suggests how the polypeptide chain is captured for translocation, basically like this. So we call it the clamp. This shows the clamp when it's still open, so the polypeptide chain would enter here, then this PPX domain would move around and close and would capture the polypeptide chain. The clamp is in fact made of conserved amino acids. So here we've colored in blue the most conserved and yet red the least conserved residues. And as you can see, the interior of the clamp is made of conserved residues. And these residues are also pretty hydrophobic, as shown down here in yellow. So this suggests that this clamp is also interacting with signal sequences and that there is not a separate binding site for signal sequences. The signal sequences would simply have a preference to bind to the clamp simply because it's hydrophobic and they are hydrophobic. So, and this would allow translocation to start at the N-terminus. The most important point here is that the clamp sits right on top of the channel. So if I take away in the side view the PPX domain, you can see that the hypothetical polypeptide chain would move from the clamp right into the SecY pore. And so what it means is that you capture the polypeptide chain and you hold the polypeptide chain right on top of the SecY channel. You position it right on the right place. Okay, let's talk about changes in the conformation of SecY. They're also kind of interesting. So first of all, the N-terminal half doesn't change much at all, but the C-terminal half does. TM7 moves, TM8 moves, TM9 moves, and this E arm also moves outwards. All these changes seem to be dictated by the insertion of this two helix finger into the channel, because otherwise you would see clashes. The channel, the plug also moves, but the plug is still blocking the channel. So as we predicted, this is not yet the open state of the channel. This is still a closed state that would not allow even ions to go through, let alone proteins. So the signal sequence is required to displace the plug completely. This is the most interesting change that we see. In the lateral gate, there is an opening. We call it the window. What you see here in yellow is actually the backside of the channel. So you're looking through the lateral gate towards the backside of the channel. And there's seams on either side of this window. On the cytoplasmic side, it's a little shaky because it depends on the position of a single amino acid side chain, and at 4.5 angstroms, we can't be absolutely sure about that. But on this side here, it's clear that there's no that the window is closed. So the distance between side chains is approximately 5 angstroms. So what does that mean? So what we think is that the channel is basically prepared for the arrival of the signal sequence. When the signal sequence comes in from the cytoplasmic funnel, it would insert into this into the window. So it would require a little bit of widening of the lateral gate, and then the hydrophobic part of the signal sequence would insert and could contact the hydrocarbon chains of the phospholipids. And this is actually known that the single sequence, hydrophobic sequence, contacts the lipid. So if we assume that the window would be maintained when the channel is completely open, and this is a total speculation, I have to admit, then this might suggest a mechanism for membrane protein integration. So here's the idea. So if you have a polypeptide chain that's moving through the channel, if the window existed, every polypeptide chain could taste basically the interior of the lipid bilayer. And if the segment inside the channel is hydrophobic enough, it could move out sideways into the lipid phase and become a membrane anchor. This would require that the seams on either side of the membrane would open briefly to allow the polypeptide chain to exit sideways. But the important point is that the seams would normally prevent the head groups of the phospholipids to move into the channel. So the channel can be stable even despite the fact that you have a lateral opening in the walls. One thing that we did not expect from the structure is that there is a perfect alignment between the mouth of the clamp, which is this interface between the PPX domain and the MBD2, and the lateral gate in the SecY channel, as shown here. What does that mean? So I think it suggests that the SecA molecule may be involved in membrane protein integration. So imagine a polypeptide chain that arrives in the channel. This is a transmembrane segment, and we're having a top view here. So this transmembrane segment would exit laterally into the lipid phase. The following cytoplasmic domain of this protein would not be able to exit unless the mouth of the clamp would be aligned with the lateral gate of the SecY channel. So what we believe then is that this arrangement allows for the simultaneous lateral release of the polypeptide chain from both SecY and SecA, suggesting that SecA is involved in membrane protein integration, which has not been seriously considered. There's some evidence, but I think this actually warrants more investigation. So finally, I wanted to address the most important question, namely, how can we learn something of how SecA moves polypeptide chains through the SecY channel? And this brings me back to this Two Helix Finger that pokes right into the cytoplasmic opening of the SecY channel. So this just shows that the channel, this Two Helix Finger rests in between the 6-7 loop and the C-terminus of the SecY molecule. And so what we believe then is that the polypeptide chain would move from the clamp and pass by the fingertip, that is the loop between the two helices, and then on into the SecY pore. So we've tested this idea by using disulfide bridge crosslinking. And the way we do this is in the following way, and this is work from Carl Allenson in the lab, who is just graduated, he's a student. So we take a substrate, probe A is a substrate, and we put the bulky tRNA at the end to prevent full translocation. This prevents the C-terminus to move through the channel. And so we're generating a translocation intermediate. And then we have two cysteines in the substrate, one to cross-link to the pore of SecY, and the other one we're going to move and ask the question whether we can also cross-link to SecA at the same time. To make a long story short, if we take a substrate like this, we can cross-link the cysteine to the pore residue here, the one in yellow, and then if we move the second cysteine up, we can cross-link to these residues shown here in color, which are exactly in the loop between the two helices. The distance from here to here is about 7 residues in the substrate, and from the structure we know it's about 20 angstroms, which means that the polypeptide chain is pretty extended when it moves from the fingertip into the pore. So the next question is then, if this loop is so important here, or contacts the polypeptide chain, is it actually important for translocation? So another student in the lab, Stephanie Miller, who was actually a rotation student, did an alanine scan. So we changed every residue in the helices and in the loop to alanines. And what's interesting is, in this loop, the only residue that is of importance is this tyrosine here. If you mutate it, you're down in translocation. There are other residues in the helices that are important, but those can be explained by maintaining the helices or interaction with SecY. But this tyrosine has no contact to anything in our structure, so likely it interacts with the substrate. You can change the tyrosine to other residues. We actually changed it to all, well, actually, with one exception. We changed it to 19 amino acids, and it turns out that you can change it to other residues that are bulky hydrophobic residues. The histidine is likely uncharged when it's functioning. And you cannot change it to small residues or hydrophilic residues or charged residues, then you're dead. So now we can go back to the database, and amazing number of SecA sequences are available. This reflects, of course, the number of bacterial species that have been sequenced. It turns out that 80% of all SecAs have indeed a tyrosine at this critical position, and most other SecAs also have bulky and hydrophobic residues, which, according to our mutagenesis, would also be expected to be functional. However, we found 18 SecAs that have small or hydrophilic residues that, according to our mutagenesis, shouldn't work. Well, that's a big, initially we thought, oh my God, we are wrong. However, it turns out that in all but one case, there is always a second SecA molecule in the same organism. So these SecAs with a non-conventional residue at the critical position are called the SecA2 molecules, and the other ones are called the SecA1 molecules. Turns out the SecA1 molecules always have the canonical residue, a tryptophan or another bulky residues like a methionine in this position. The SecA2 residues have these unusual residues here, and it turns out that SecA2s transport only one substrate in the cell. These are long polypeptide chains, adhesin molecules. These are all pathogenic bacteria. They are serine threonine rich, and these substrates are actually glycosylated in the cytosol before being translocated. Whereas SecA1 is responsible for the translocation of every other substrate in the cell. So this is very special. And we have done mutagenesis, and it's clear now that this SecA2 molecules are basically inactivated for translocation of the normal substrates by this mutation. For some reason, they're not inactive to transport this particular substrate. But it seems, I think, a very good piece of evidence that this tyrosine is in fact really an essential residue, because we can pick out the SecA2 molecules by just looking at one residue. Of course, there are many other mutations and differences, but we can pick them out by just looking at one position. So the model is then that the polypeptide chain moves from the clamp, passes by this fingertip. The tyrosine is the most important residue in the contact here, and then it moves on into the SecY pore. And so this suggests then a mechanism in which this two helix finger would move up and down and push the polypeptide chain into the channel. So the idea is that in the ATP state, the finger would move down, and coordinated with this movement, the clamp would have to loosen its grip on the polypeptide chain to allow the finger to drag the polypeptide chain into the channel. And then the clamp would tighten, hold the polypeptide chain, and then the finger would reset to grab the next piece of the polypeptide chain, basically like this. So, okay. All right. So the tyrosine, we believe, makes the major contact with the polypeptide chain, and how exactly it does it is not so clear. The most likely explanation is that it interdigitates between side chains, and this would provide it with enough grip to drag it down when it moves downwards. What's really incredible is that the hexameric ATPases that move polypeptide chains may do it in exactly the same way. So there's a bunch of hexameric ATPases like the CLIP proteins, the P97 ATPase, 19S proteasome, the FDSH proteins. In all these cases, 6 subunits come assemble in a rings arrangement. The pore would be in the center of this hexamer, and each of the subunits has a loop that points into the central pore. And the idea is that this loop would move up and down, pushing the polypeptide chain to the central pore. And it turns out that in all these cases, there's a tyrosine at the tip of the loop. And the tyrosine is the only residue that matters in this loop, just like in the SecA case. There's, in the case of P97, it's a tryptophan, and FDSH is a phenylalanine. But we've shown in our mutagenesis that those residues are working just the same way. So what we have here is a very conserved general mechanism by which polypeptide chains are moved by ATPases, basically always with this kind of loop and an aromatic residue at the tip of the loop. And this brings me to the end of my talk, just acknowledging the people who done the work. Andy Osborne did all the decipher bridge crossing experiments. Jochen Zimmer did most of the work on the structure. SecA SecY structure with help from Yunsen Nam, and Carl Allenson and Stephanie Miller did the work on the cross-linking experiments and also the mutagenesis. Thanks.
Tom, is this on? I have a question about the clamp.
About
the SecA clamp.
Yes.
So, according to what you said, that is largely hydrophobic. So I can see how it would bind to a signal peptide and then hand that over to SecY. But the bulk of a pre-secretory protein is hydrophilic. So
Right.
how would the clamp then engage successive segments of a polypeptide and hand them over to SecY?
Yeah. So the binding of SecA to substrate is actually pretty weak. So if you just take SecA in solution and add substrates, it's very hard to detect their specific interactions. So my suspicion is that the hydrophobic interaction isn't actually very strong.
You provided a very nice explanation of how the moving tip of the two helix finger can help push the polypeptide chain into the pore. Do you have any idea what keeps the retracting tip from pulling it back out? Because it's a usual problem of any molecular machine.
So this is what we believe with a clamp. I mean, I should say that in all these ATPases, you need more than 2 states to do translocation. This is true for all these ATPases because, as you say, if there was just 2 states, you would reverse the movement that you just did by moving backwards. So there needs to be at least 3 states, and those could be the ATP state, the ADP state, and the nucleotide-free state or the phosphate release. There are several possibilities. But in any case, what we believe is that while this finger is resetting, you hold on to the polypeptide chain with a clamp. So there would be a coordinated movement. This is speculation. We haven't actually even shown that the polypeptide chain moves through the clamp. This is certainly something that we just actually doing right now.
Tom,
I was really intrigued by the variant SecA-18, the variant that was found in the pathogenic bacteria. So in those cases, the presumption is that they've changed to a more hydrophilic residue, maybe to accommodate the carbohydrate chains. And if that's true, does it have implications for retrotranslocation of antigenic peptides?
Oh, well, it's two different
related questions, yes.
Yeah, two different questions. So on the SecA2, we've done mutagenesis, we meaning this is a collaboration with Barbara Bensing and Paul Salem in San Francisco. And so if you change the hydrophilic residue to any other residue, it doesn't actually abolish translocation of this special substrate. And it's to some extent even translocated even without a carbohydrate chain. So I don't think it's necessarily the interaction with the carbohydrate chain. It goes better with the hydrophilic residue, so there is some effect, but it's not the only explanation. So the second question is retrotranslocation?
Yeah, so because it's known that antigen processing of glycosylated peptides is quite distinct, and so I'm wondering if you could speculate on whether you'd expect a variant channel for glycosylated peptide.
I don't necessarily expect that because, but the evidence that the SecA-61 channel is actually involved in retrotranslocation is, I'm obviously in part responsible for this, but Yihong actually had the best evidence that it wasn't really true. Whether that's there's still some nagging doubt that maybe the SecA-61 channel could still do some retrotranslocation, but I think the evidence is pointing against that mostly.
So, would you like to speculate about how polytopic integral membrane proteins might get folded up in the membrane?
Okay, so the question is how are polytopic membrane proteins integrated? Now this requires a little bit of a longer answer, and I would need a board probably to explain it really well. But basically, the simplest model is that the first hydrophobic segment, this is the first point, the first hydrophobic segment would determine the orientation of the polypeptide chain. And then every other hydrophobic segment that follows would follow that topology. So for example, if the N-terminus is in the cytosol first, then the next one has to have the opposite orientation and so on. So from the structure, it's clear that you can have at most one hydrophobic segment in the lateral gate and one hydrophobic segment in the channel proper, no more. And so the idea is that one segment, or at best two segments at once, could leave laterally into the lipid phase. So basically, if the first hydrophobic segment, and then the polypeptide chain would alternately be translocated to the other side of the membrane or into the cytoplasmic side, depending on whether you precede or succeed a hydrophobic segment. I can draw you some schemes later on if you want.
Tom, the cross-linking data suggests that the SecA contacts the SecY that's non-translocating more tightly than the one that's translocating. But in your crystal structure, of course, you crystallized it with the presumptive translocating copy of the SecY. So what do you think changes during translocation to reorient?
No, I'm sorry. What are you saying?
So the SecA is presumed to bind more tightly to the non-translocating copy of the SecY
I see what
dimer.
you're saying. Okay. So we've shown that the only way we can get a stable complex of SecA with SecY in detergent solution is by using ADP and beryllium fluoride or aluminum fluoride. And this is the state where the finger is the deepest into the channel. So we think that this allows the strong contact of the translocating copy with the SecY channel, of the SecA with the translocating SecY copy. If you look in membranes, you get relatively weak interactions, but in this case, we see much of the cross-linking to the non-translocating copy.
I have a little announcement. For those who prefer to talk to Tom in a more private setting, we have a reception after this seminar, which is down in the hallway. Thank you all for coming.